Good night, good afternoon, ladies and gentlemen. Here we are with another science program. Are you ready? So let me start. Okay, so this is, let me make it full screen. Analysis of photosynthetic bacteria. Actually, let me go back to regular screen and make it bigger. I think it works better that way. Okay. So name of the talk is analysis of the photosynthetic bacteria. Again, this is a homework. This was a homework from the class experimental microbiology, and I think I did this back in 2007. So it's an important topic, and I believe it is still important. So photosynthetic bacteria. So let me just introduce some of the facts. As we know, life on Earth was without oxygen at the beginning. There was no oxygen in the atmosphere. So and with the help of photosynthetic bacteria, the oxygen production started, and again, like the organelles, cellular organelles called chloroplast, which scientists now believe were a bacteria millions of years ago. So what is photosynthesis? Actually, what kind of an organ or organelle is needed to carry out such a function. I mean, like photosynthesis. Everybody knows the word photosynthesis, but when it comes to understanding, the proteins and the pigments and the reactions and everything behind it, it is very complex, and it starts from the bacteria, to understand. And we have to understand the cellular structures to understand photosynthesis, and especially membranes, cell membranes, and membrane proteins. Remember, proteins are made up of amino acids, and amino acids vary, among them. So some of them are like hydrophobic, and some of them are hydrophilic. So when they combine together to make a protein, and some parts of the protein becomes hydrophobic, and some parts of the protein becomes hydrophilic. And so hydrophilic means soluble in water, and hydrophobic means soluble in lipid materials or membranes. And as we know, cell is made up of a membrane. It has a membrane. Cell. That's the whole point, actually. So what is cell membrane? I mean, is it just a lipid structure? It's a bilipid layer, or, so what does it do? To understand is not easy. Why? Again, I'm gonna go back to the pandemic, the COVID pandemic. Even the vaccine was produced in a way that it was encased in a lipid material, liposome they call it. Even most of the creams and all the beauty products these days are synthesized and packaged in liposomes. So what are liposomes? It's just like membrane envelopes, like viruses. So 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 so. so Again, now with today's technology, we are using these membranes for better delivery of drugs, for better delivery of medicines and creams and vitamins and etc. So 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 this is kind of related to that, to understand what's inside the membrane, what is a membrane, and it's a workspace. It's a kind of a bench, and same thing for mitochondria. It is all the vital reactions, like we are now utilizing oxygen. Still, it takes place on the membrane structures of the mitochondria. Plants generate oxygen with the membrane structures, and bacteria does it and did it millions of years ago, billions of years ago. So let me just start with this introduction, and then I will try to summarize it as much as possible. Again, this is a very good topic, and it was a very good experiment for us. It was very educational, so I just wanted to share it with you. Introduction: environmental signals stimulate bacterial pathways. Response result in synthesis of required metabolites and gene products. Bacterial genes are regulated by different responses and stresses. In this experimental photosynthetic bacteria, Rhodospirillum rubrum, will be analyzed under a semi-aerobic condition, which will stimulate production of photosynthetic apparatus and corresponding photosystem pigments and proteins. Just so one part of the experiment was just to stimulate the production of photosynthetic apparatus and corresponding pigments and proteins. Remember, this is like germinating a seed. A seed is not green; it doesn't have roots or anything. But once you soak it, and it starts to develop, and it starts to produce these photosynthetic apparatus and photosystem pigments and proteins. And that is true for a plant, for a seed, like a bean or something else. But same thing applies to the bacteria, photosynthetic bacteria, and to understand the system, it is easier to study it in bacteria, which is a lot easier to study. Oxygen regulates the synthesis of photopigments and the pigment binding protein of the photochemical complexes. See, it's getting complex already. See again the word complex, photochemical complex. So in the presence of oxygen, the synthesis of photopigments and the pigment binding proteins of the photochemical complexes. So I mean the stuff that we eat, the sugars, the oxygen we breathe and the sugars we eat, all depends on photosynthesis, and then how it started is important. So this experiment kind of studies that. Regulatory response of the cells can be assessed by evaluating pigment content via spectrophotometer. That is just pigment content, like carotenoids and etc. Uh, is induced and in induced and uninduced cells. That's just the word for the cells that are ready to synthesize these pigments. Once you induce the cells, then they start making these photosynthetic elements and proteins and pigments. So it's called induction, induced and uninduced cells. So this is very important, because it's like a seed. If you keep the seed in the package, nothing will happen. Nothing will happen. But if you induce it, then a plant will grow out of it. So there's a big difference between induced and uninduced. So that is true for most of the life situations. Induced, uninduced. We have the same genes. Doesn't matter you have the genes or not. If the genes are not induced, then nothing happens. In addition to spectral analysis, protein composition of the cells will be observed and detected with sodium hydroxide sulfate, polyacrylamide gel, electrophoresis, along with analysis of mutant strains for their photochemical protein and pigment composition. The rest is, is kind of easy to just understand, because remember I mentioned the word complex. So it's an experiment, trying to understand these membrane proteins and membrane structures. Phototrophic cells from intracellular cell membrane. That is see that's the key sentence kind of. Phototropic cells, meaning cells that utilize light as an energy source. Phototrophic cells form intracellular cell membrane. And when there is no light, or when there is when there is no such a need, they don't form this ICM. It's called ICM, intracellular cell membrane. Normally they don't synthesize it, but in right conditions, when they are induced, they make this intracellular cell membrane. It's like an extension of the cell membrane. So with proteins in it, because now you need to synthesize pigments and other photosynthetic proteins. And their photosystem apparatus light harvesting antenna, which is like a carotenoid, that just like a chlorophyll, but it is different in this case. It's called light harvesting antenna. See, bacteria has antenna. Who would think? Light harvesting antenna. This was very interesting when I heard the word. Like light harvesting antenna in a bacteria. What are you talking about? It's like a it's like a spaceship. Having all these like different devices and gadgets and having an antenna on a bacteria. So I was like amazed, like what the heck is going on? And the reaction center, because once you harvest, see the very important light harvesting antenna. So we're talking about energy here, because you need energy, you need photons and some sort of energy to carry the photosynthetic reactions, and then it starts with light harvesting antenna and reaction center, and then you have to turn the photons into electrons and then make ATP. That's the whole point, that's just the whole point. The rest is details. Okay, don't get too drowned in the details, but just get the idea. Even same thing applies for the plants as well. I mean, it's just the chlorophyll is the antenna in plants, the green stuff. The chlorophyll. Chlor in Latin language means green, and philic means green, liking, and so that makes sense. Bacterial chlorophyll. See the word bacterial chlorophyll, BCHL, is found in light harvesting unit of the system. So see, that's the bacterial chlorophyll. Aerobic grown cells do not display intracellular cell membrane (ICM) in the presence of oxygen. They don't make ICM. So this is one thing to remember. In other video, there's cell regulation. In bacteria, they have DNA as well, and on DNA there are genes to synthesize proteins and etc. But those genes do not get turned on without necessary signals. And then here is an example. Aerobic means if oxygen is present, the cells in this particular bacteria do not make intracellular cell membrane. That houses these photosynthetic elements. Aerobic grown cells do not display ICM formation and its components. See, cells make photosynthetic components when necessary. Bacterial cells in this case. Light harvesting antenna and reaction center are found in close proximity on cell membrane, thus providing an electron transport system resulting in a proton hydrogen gradient, which can be used for production of ATP by ATPase that exist on bacterial cell membrane. See, all these structures are present one after another. It's like a cascade of reactions. You capture photons and turn the photon into electron and process those electrons and then deliver it to the ATPase, and then you get ATP at the end. That's the whole idea. And with the help of ATP, you synthesize glucose and just bring all those carbons to make a sugar, to make glucose, and that's what life is all about. I mean, the life starts with glucose, just basic. And then to synthesize glucose, you need ATP, and to have ATP, you have to have ATPase running. The ATPase needs energy, and then there is a proton gradient. That's how it works. This ATPase. If you like Google it, you will see how ATPase works, and then basically it's like a battery. Or a dynamo or something like that. It works with the flow of protons. So electrons are transferred from a light harvesting system to reaction center via P87 electron carrier, then relate to the quinine pool and recycle back to reaction centers with iron sulfur bound proteins. It's not easy, but just to make it simple, you have to kind of recycle these electrons. I mean, once you capture the photon and turn it into an electron, and now you have to process it with electron carriers, all located on the membranes. All these parts and components are located on the membrane, cell membrane. RC reaction center is composed of three subunits encoded by two different genes, POF-M, POF-L, and POF-A. So two different genes synthesize the three different subunits, which make the reaction center. That is needed to process these electrons so that you can generate ATP, which encodes respectively for ML and H proteins. Okay, so H subunit of the reaction center is essential for functional reaction center assembly. So I said that the H subunit kind of keeps them all together. Light harvesting unit, however, consists of alpha and beta subunits. Remember from the yeast to hybrid talk, all these proteins, sometimes they have subunits and different domains, and in this case, it has alpha and beta subunits. The proteins sometimes are combined and to carry a function, which are encoded by POF-I and POF-B. It's just now talking about genes. Aim is to characterize POF and POF mutants for their ACM and photosynthetic system components. What happens, if you delete these genes, like all different subunits, and what is completely necessary, what is not, and it's like one of those mutant studies. You delete them and see what happens. And in this case, it is about those genes and their function on the ICM. ICM formation cannot be completed without POF gene products. Plasmid pH 3.6 plus POF-A restores H15 RC mutants which lacks POF-A gene, and then how you work with these mutants. You insert a plasmid carrying the right gene, and then you restore the mutation, and so you understand how it works, and etc. But flanking regions and gene is replaced with a kinase cassette, and sometimes depending on your lab, and the flanking regions means if you just copy the gene, that's gene by itself, but oftentimes, remember, genes have control units, oftentimes located at the upstream part of the gene, and it's called flanking regions, in a way, kind of shoulder of the gene, and so these are regulatory regions. So in this case, so when you define a mutant, you have to tell, does it have the original promoter, does it have the native promoter, or or what is missing? You have to define it clearly, so that others can understand, what kind of mutation is it? It is just missing the gene, or just missing the entire area region. Including promoter and regulatory regions, etc. Because sometimes, if you replace the gene only with something else and keep the promoter, and you can do other, experiments, and etc. H15 mutants do not have RC pigments, and so membrane fractions are deficient in absorbance at 800 nanometers. Again, defining mutants and what they do and what they don't. And this is membrane fractions of these mutants also display low absorbance at 880 nanometer compared to wild type. absorbance at 880 nanometer corresponds to existing light harvesting components and pigments in strains. In the wild type and in regular cells, if they have everything, all the pigments and proteins, they absorb, this wavelength very well. Nanometer, 880 nanometers. So, which is closer to, I think, the UV, which is closer to the UV wing. Under oxygen limiting conditions, this is important, the repression of PUF and PUF genes occurs, therefore inducing assembly of light harvesting system and the reaction center around cell membrane by invagination and proliferation of ICM. And so there are more than one unit of ICM. I think of ICM as a system, photosynthetic system. Like even in regular plant cells, there is not one chloroplast. There are many of them. And the same thing for bacteria. If they want to synthesize, ATP from light, and then they make more of these genes so that they can make more ICM and then generate more energy and utilize light. But under oxygen limiting conditions, derepression. Derepression means if the gene is under pressure, it's called depressed. The derepression is the release of the gene. You release the gene, it means derepressed. So derepression, it's a term, that means gene is turned back on. It's not repressed. Repressed means it's like the gene is turned off. Derepressed, gene is turned on. So under limiting conditions, when there is not enough oxygen, they just because now the oxygen is used for what? Used for generating energy. And if there's no oxygen, how are you going to generate energy? So the cell employs this system of generating energy from light under oxygen limiting conditions. The whole idea of utilizing oxygen is to generate the energy. Oxygen is the final electron acceptor in our mitochondria. Is the final oxygen acceptor. So the cell makes this structure ICM under such conditions. Strains can be stimulated for ICM formation via expression of heterologous membrane proteins, which is lethal to host cell as well as causes formation of inclusion bodies in the cell. And then this experiment is actually about another topic, how you can induce formation of ICM. Is it possible via other routes and via other systems or methods? If you express heterologous, like some other protein in the cell, can you stimulate ICM formation? So the second part of the experiment is about that. So deletion mutants can be complemented to restore ICM formation. Again, you complementing means you bring the gene back, and then restore the mutant, and then restore the system and the photosystem function. When POF and POF genes are expressed on a vector, meaning plasmid, host vector system is widely used for production of recombinant membrane proteins, expression of open reading frames, assays, and physical analysis of proteins, as well as commercial membrane protein synthesis. So this experience is important. Why? Because it is about production of recombinant membrane proteins, and even in clinical studies. Remember the virus, the virus, it had a spike protein. Remember, and then they built the vaccine based on the spike protein, and then spike protein was binding to something in our cells. Remember, it was the ACE protein on our cell membranes. Remember, that's how the COVID virus, that's how it was getting into our cells. The virus was connecting to our membrane proteins, our cell surface proteins. So that's why this experiment kind of is important, because a lot of labs that are studying membrane proteins, they want to synthesize and generate these proteins, and the membrane proteins, they're kind of harder to synthesize, because they're not. Remember, membrane is mostly filled with lipids. It's a lipid structure, and therefore the proteins that are found in membranes are slightly different than ones found in cytoplasm or cytosol. So it's a bit challenging. So it says, that the host vector system is widely used for production of recombinant membrane proteins. So if you can trick the bacteria to make these membrane proteins with the help of ICMs, the cellular membranes, remember, so ICMs. So it's a kind of the advantageous. It's kind of advantageous to use bacteria to generate or produce these membrane proteins. They're important in many clinical studies. And physical analysis of proteins, as well as commercial membrane protein synthesis. So let's continue. Just to give you the idea, like I said, again, don't be too bothered with the genes and their names and strains and plasmids, and now it's gonna be a lot of details. So I will just and let me see how many minutes so far. Let's see, half an hour already passed by. So there are, I think, 2 strains here. So M1 strain, there is this first strain which lacks the POH gene and related areas, flanking regions, and the H2 strain with a POH deletion and associated regions, and another deletion, POF operon. So H2 strain is also missing the POF operon. So it's a kind of a double mutant. Will be used for analysis for induction and expression of recombinant proteins. Flanking open reading frames, deletions, and ensure removal of native promoter. So just they are, like I said, there are different deletions. Sometimes you just delete the protein, section of the gene, and sometimes you just delete the whole thing, with the promoter region, the native promoter region, to stop expression of downstream genes or whole operon. Because in the bacteria there is this system called operon, so it has a one switch box that turns on the protein synthesis for many genes. So depending on the mutation, sometimes you have to delete the native promoter. I think they did that. To kind of stop expression of the whole operon. It's the assembly line kind of in bacteria that these proteins get synthesized in an assembly line, and there is usually one switch box, and then you have to sometimes control that as well. And then second part, vector PRX will be used to express Pseudomonas, MSL, and cytochrome B proteins with an oxygen-regulated promoter in strains H2 and M1C. You just wanna express or produce these proteins, the MSL and cytochrome B proteins in strains H2 and M1 with the oxygen regulating promoter, and then just with a plasmid. Vector has a multi-cloning site and a terminator fragment. Okay, so it's just describing the plasmid. Here we will screen analyze fractions from differential and standard centrifugation. So that is the important part, the centrifugation part, because to get the proteins out of the membrane is kind of a challenging process. I mean, like this. I think I remember we have used, everybody remembers the French press, what makes coffee. It has a kind of a filter, and then a filter piston, and then you just and then you press the coffee, and then juice, that stays on top. So I think we did we tried some methods, and so we will screen and analyze fractions from differential and standard centrifugation, standard and differential centrifugation. I think we used different high power centrifuge, and I remember professor saying we respect this device, and I think we cooled the samples, and then so that they crack open. I think because extracting stuff from membranes is really hard. So I think we froze them, and then it was a super high power centrifuge. And I remember, because to start with, I mean, to everybody, whoever is listening, if you are working with centrifuge, you have to be very careful. You have to respect, because if anything is off, I mean, if it is a high power centrifuge, and it's like loading a washing machine, and still people do that. Even if your car is making sounds from the tires, if the tire is unbalanced, at high speeds, you will hear all types of different sounds from your car if it is unbalanced. So same thing with the centrifuges. You have to treat them with utmost respect. they make like thousands of rotations per minute. And basic, I mean, DNA the centrifuge is like I remember it was like 12,000 rotation per minute. I mean, 12,000 RPM. It's called RPM, rotation per minute. And this was like much faster and much stronger than that. It was a big centrifuge. I remember. I mean, it was in this special room, and then so and then just extract proteins from these bacteria and bacterial membrane. I mean, like extracting proteins can be easy, can be hard. See these are cells R5 wild type, M1 PUHE deletion, H2 PUHE and PUF deletion. See, H2 is a double deletion, M1 is a single deletion. And look for protein composition on the STS page, Western blotting, and survey for the chemical components via spectrophotometer. Just survey the pigments with the spectrophotometer. And then these are the recombinant protein, the MSL and the cytochrome B production of H2 strains will be tested. If like this is, remember, this is regarding production of recombinant proteins, membrane proteins, and tested with the immunoblotting method. Pigment and protein content of cells under limiting conditions will be assessed and evaluated among different strains and cultures. Like even like you go to markets and these pigments like beta carotene. I mean, anything you buy, even the sodas and everything, they have beta carotene. They have pigments, and how do you think they make them? How do you think they just produce them? How do you think they make beta carotene? All these carotenoids, and they use cells, and they use cells, and sometimes algae and other organisms. They use these cells and bacterial cells, algae, to synthesize. Even agar mostly comes from the algae. So the plants, I mean, algae is a plant, and now scientists and many of the industrial systems utilize algae to make even biodiesel, oil, and then various other stuff, including membrane proteins, pigments like carotenoids and beta carotenes, and these are antioxidants. Antioxidant carotenoids, stuff is that gets the name from carrot, so pigments, and will be assessed and evaluated, and etc. So let's just go back to, I think this is the first table that we have tested the pigments, the first table. So it's a long, but see a bunch of numbers, and then just wanna show this is the absorbance peak. So this is the wavelength. So there's pigments from various samples. So this is wild type, and induced and controlled. In control means not induced, and then wild type induced. And then if the 186 means it is ultra centrifuged. So it means it is ultra processed. Remember, it is hard to get these proteins dislodged from the membrane. So this specimen 186, if it is saying 186, it means it is ultra centrifuged. So that you have more protein, more extraction. Like I said, it's a better because you have to put you generate mutants, put the plasmids, and generate all these proteins, and then at the end you have to extract them from the membrane. And in this chart, you see, even with the control, see highest one. This is control R5 with high, centrifuge, with ultra centrifuge, has the highest amount of pigment, highest amount of ICM formation induced, and ultra processed, and then you see, and then next one, and the next one is see, this is the, remember, first strain, with single deletion. You get the, you still get this is the gene. Remember, this is mutant, but it has it has the gene for the antenna, and a light harvesting unit it still has the light harvesting unit, but not in a reaction center components. So M1 with ultra centrifuge, that is the second high peak, 0.3, and then this is the control. So it's a kind of, and then if you look at other numbers, they're not like much. As you see, this is like just plasmid control, with no gene on it, and not induced or not properly processed. If you cannot get the the pigment or the system out of membrane, it's no good. So methodology is important. So that's kind of shows. So this is like first mutant, second mutant with control, and then with different centrifuge speeds and systems. Standard centrifuging and special centrifuging. So, like I said, this is just this was one of the first results, from the experiment. And the yellow one is the R5 induced with the regular standard centrifuging. With standard centrifuging, you still get some peak. That means there are pigments that are absorbing this wavelength, 880 nanometers wavelength. It's just a photosynthetic wavelength. It's just so. What these colors refer to. It's a photosynthetic wavelength. It's starting from 400 and all the way to 900. And this is the in case of wild type, and this is the case, as you see the numbers. So I'm just gonna. So this is the analysis of pigments absorbance. Analysis of pigments. So this is STS page analysis for protein composition. Remember, there are proteins that carry out these functions. The electron transport proteins. These are called electron transport proteins. And this is the molecular weight, 32 kilodalton, and then 16. This is lighter, and this is slightly bigger, almost twice as much. And you in number 5, you see the full system, with all the components, and this is again like induced, not induced, and in regular centrifuge and ultra centrifuge. Remember, this 186 means like a fast centrifuge, and R5 is wild type. And H2 is the H2 is double deletion, and with plasmid and etc. So as you see, so we see proteins here on number 9. So the STS page, well, number 9, you see proteins here, as you see, the different ranges, like 20, 24. These are proteins, the photosynthetic, ICM proteins here, as you see. So like 1, 2, 3, 4, at least 4 bands I can see, and 5 bands, that's just the photosynthetic system. Proteins. Number 9 is just with the high centrifuge you are able to see these proteins, even if you restore with the mutants. See, remember, this was a double mutant and restored with proteins and genes, and as you see on number 9, you see all of them as good as number 5, which is wild type control induced. So number 5 here, you see all these nice bands, all these nice bands, and then number 9, you see all these nice bands. This is a mutant restored, and you see all these photosynthetic components and parts and genes, and this is all proteins. STS page analysis for protein composition of these, mutants and wild type. So I'm just gonna, and then again for the recombinant proteins, remember that production of these recombinant proteins are important, because most of the clinical studies, most of the medical studies are carried out with recombinant proteins. So same thing here. H2 was a double mutant. So the first line is molecular marker, starting from 16 kilodalton and all the way to 80 and even 110. Number 5, we see a great band, and number 9, for some reason it worked better for number 5. This is see, it was this strain, this mutant strain. It had a double mutation, and it was complemented with a plasmid induced limited oxygen conditions, and you get a nice protein. You generate protein, and this was an experiment to show the the recombinant gene production. And number 5, it showed a nice dark band, STS page band, and number 9 again, we saw same plasmid and the same strain with double mutation induced, and and there's some blank wells in between. So let's continue. So discussion, getting closer to the end. Fractions of differential and standard centrifugation solutions were analyzed via spectrophotometer for protein and pigment content. Results display significant differences between induced and uninduced wild type R5 strain, as well as slight difference between, even though I typed slight, but there is difference between the ultracentrifuge and standard centrifuge. Ultracentrifugation enabled better differentiation of protein samples. See, because it's just you are able to pull the proteins, better differentiation of protein samples and structures like ICM, allowing detection of photosynthetic proteins via spectrophotometer. Whereas standard centrifugation was not sufficient to test actual presence of such components. In this experiment, we find ultracentrifuge sample 186 data more reliable for analysis. Therefore, discussions will be based on 186 samples, unless otherwise stated. P18 data is also shown on table 1. This is standard centrifugation. Standard centrifugation. So it's in all medical experiments or in all cellular experiments. I mean, you just you have to extract the protein. I mean, you just so extraction is a problem in these protein types and membrane proteins, because they are kind of embedded in the cell membrane. They're kind of just hard to extract. It's kind of mining, because all sorts of mining is hard, and this is kind of protein mining, to be honest. I mean, if I have to say in regular words, this is protein mining. You have to dig it out of the membrane, because that's where a lot of the reactions take place in the cell, among the membranes. These proteins are embedded in the membranes. We definitely see presence of 500 nanometer absorbance of carotenoids. Like check. I dare you, check all the packaging, food packaging. You will find carotenoids, beta carotene, and they are bound to proteins in most of the cases. Which we definitely see presence of 500 nanometers. There's kind of a red wing, on the red wing, absorbance of carotenoids. That's why they are orange, like carrot, which give reddish color to induced R5 wild type phenotype when they are induced, in semi, oxygen levels. In addition, we also know that this wavelength is also usable for photosynthesis. So this happens, of course, in bacteria, but this wavelength also applies to plants and photosynthesis. We observed several absorbance peaks of these carotenoids around 0.17, 0.12, and 0.6, which suggests the presence of different pigments. These are different peaks, and if there are other pigments, and then you see their peaks. We see lower levels of absorbance at the 650 and 7. This is mid-range. Because remember, why plants are green? Because they reflect the light, the green light. They are green because they don't utilize the green light. For some reason, they reflect the shades of green. For some reason, we don't know. Just to see, it's the same thing for bacteria as well. Even though these are reddish, kind of. Even in the fall, remember, in the fall what happens? The chlorophyll is broken down, and then you see other pigments in the cell. The yellows and oranges and reds and everything. That happens during the fall. There are other pigments. I mean, the plants utilize all kinds of pigments. To carry out photosynthesis, and lower levels of absorbance at 650, 700, and 800 nanometers, and peak at 880, which provides evidence for light harvesting antenna pigments. That's where they operate at this. Excuse me. At this wavelength, 880 nanometer wavelength, and the pigments, and they capture the energy. I mean, it's a high energy light. And R5 control strains were not induced They're like ICM. I mean, if you don't trigger the system, so they don't produce these proteins in the system, the ICM system, and the photosynthetic proteins, thus causing absence of peaks of, 500. I mean, if you don't provide necessary signals, I mean, the cells will not make necessary components, and then you will not detect them. So induced R5 cells and induced M1 strain profiles displayed similarities for 500 nanometer absorbance of carotenoids and 880 absorbance of light harvesting antenna proteins. However, M1 induced cells, this is with single deletion, lacked peaked peaked at 800 nanometer for RC center proteins. So this one missing one protein of the reaction center. This data was also supported by STS page results. Absorbance of M1 induced 186 samples started at 0.1 at 400 nanometers, which is lower than R5, and followed peaks at 0.1 at 500 nanometers, lowering to 0.04 at 600 nanometers. So that was the mutant. Overall peaks were lower. Overall peaks were at lower absorbance compared to control. This was control R5, and with high centrifuge extraction. M1 strains M1 was a single deletion. M1 strains were lacking PUA gene, and here we see induced but RCA deficient phenotype. So it was missing the components of the reaction center. H2 strains were deficient for PUA and also the PUF operon. Absorbance of H2 induced cells did not display peaks for reaction centers at 800 nanometers because they are missing all these proteins. So they didn't display the peak at 880. Therefore, data for control and induced H2 strain was quite similar to R5 control cell profile. I mean, so the wild type acts like a mutant when there is no induction. If the proteins are not necessary. If the cell is not induced, it will act like a mutant. I mean, it will not synthesize those proteins and pigments, and very different from M1 induced and R5 induced cells profile. STH page assays for these strains separated cell proteins as Western blotting. STH page assay, another name for Western blotting. Wild type induced sample lane. See, this is the wild type sample lane. Showed reaction center protein H 27 kilodalton, protein L 30 kilodalton, and M 34 kilodalton. See, those are photosynthetic proteins like H, L, M. There's just three of them. However, M1 induced sample displayed only LH proteins, which formed the dark band with other low molecular weight proteins. So M1 was with the single mutation. It showed only the light harvesting proteins that are bound to pigments. So light harvesting, M1 induced sample displayed only light harvesting proteins. single mutation. With other low molecular weight proteins, there are two light harvesting proteins. Light harvesting alpha protein, 7.1 kilodalton. That's a small protein. Another one, light harvesting beta, 7.7. Light harvesting center proteins could not be found on all strains of H2. So this is a double mutation, missing the reaction center. Reaction center proteins could not be found on all strains of H2M1 and uninduced cultures when compared to reaction center proteins of wild type induced sample. Light harvesting proteins were only observed on lane 5 and 10, which host the intracellular membrane and a light harvesting antenna, which I showed on the table earlier, lane 5 and 10. Western blot analysis of H2 strains expressing recombinant proteins, cytochrome B and MSL from Pseudomonas species was successful. Genes were cloned on a low copy plasmid to eliminate toxicity of soluble proteins. So it's kind of challenging to produce these proteins. Specific promoter was oxygen regulated, allowing us to see the effects of mutation and induction. And host the H2 cells were lacking both light harvesting and reaction center complex, resulting in ACM deficient phenotype. But after transformation, once you transform with the plasmid with corresponding membrane proteins, we expected to detect the presence with his antibody, because if the proteins are expressed on a plasmid, then oftentimes they have tags with histidine amino acids, which you can detect with the histidine tags or histidine antibody system. So that's the one way. That's one way to to see if the protein is synthesized from the plasmid. So that's how you detect it with his antibodies. Since these clones express his-tag proteins, immobilon contain two of the recombinant protein in different amounts, yet same amount of cells were used for loading. Say when you load the same amount of same volume of cells, but sometimes one generated more than the other. They just suggest that MSL protein production was significantly higher than the cytochrome B synthesis, because band belonged to sample 5 and 9 were quite thicker than of lane 7, which shows cytochrome B data. Vector alone and uninduced cultures displayed none of the proteins, supporting evidence lanes corresponded to molecular weight of 20 kilodalton. That's how you know that's how you know the protein, by looking at the size. So there was protein synthesis, recombinant protein synthesis. Experiment was successful, and here we have obtained expected result and accepted our hypothesis that rhodospirillum rubrum photosystem is regulated by genes that responds to oxygen stress. He yet provided evidence and synthesized recombinant membrane protein production in H2 double mutant cells, which are defective in producing membrane pigment and proteins. So it's part of the experiment. So we were able to show, the recombinant protein production, and then show the system of ICM, how it works. So it's an interesting paper, interesting experiment. It's just remember. I mean, still this day, just we use the membranes, the liposomes. Remember the word liposomes. All the creams these days, even the vaccines were are now being administered. The in liposomes, uh, in membrane structures, and it's important. So this is it. Uh, there are many things to talk about membranes, but this is just the start. Like I said, the respiration, photosynthesis. Even our the diabetic people, insulin. Insulin facilitates the diffusion of glucose into the cell via glucose transporters. Again, the glucose transporters, their numbers vary, and certain food that we eat increases the number of glucose transporters. You will be diabetic if you don't have enough glucose transporters, and these are again membrane proteins. Glucose transporters are proteins that transport glucose into the cell, so that you don't your blood doesn't become a syrup. So it's important in these membrane proteins, and how they work and how they are structured. you know so these kind of experiments help us understand that you know so this is it for now. Thanks for listening, and please subscribe to my channel so I can continue talks like this. Hopefully it is helpful, whether you are a biology student, or you are a medical student, or you are a researcher or a scientist. I do my best, and forgive me if I made any mistakes, or anything that sort. This is just to give you an idea. This is again my lab report that I made. I typed it. This is my work. This was a homework. I'm just giving you an idea on these topics. Thank you very much.